Well, thanks very much for turning up. I've given talks on pitcher plants to myself and one other person on occasion, so it's nice to have plural people turning up. All right, I'm going to start it here. Just to sort of confuse things, people who know what I do are probably wondering why this isn't a talk on hummingbirds. Most of my work now at Royal Roads is around bird migration. This is a subject that I started doing for my PhD many years ago, and I've just been sort of sticking my toe in the bath every now and again, just to keep abreast of things and doing a bit of research at the same time. So this is a talk I don't normally give. I usually give a talk on bird migrations. So if I start talking about birds, please slap me and get me back on the plants. What I'm going to be talking today about is, oh shit! Thank you. I'm available for parties, bar mitzvahs, and weddings as well. What I'm going to be talking about today is about the Nepenthes pitcher plants. Actually, we'll go on this side. Is everybody awake now?
Thank you, yeah.
Is a Nepenthes pitcher plants. It's one of the carnivorous plants. There are quite a group of carnivorous plants, but the most famous are probably the things like the Venus flytraps. This is a sundew. This is a plant which uses stickiness to catch insects. This is a fungus gnat stuck on the leaf. There's a couple of groups of pitcher plants as well. This is a pitcher plant of the genus Sarracenia. This is a North American group of pitcher plants. Anybody here from Newfoundland?
I lived there for 2 years.
It's one of these Sarracenias is the provincial plant of Newfoundland, now Newfoundland and Labrador. Did you see any of those up there?
Yeah.
Yeah, they're pretty cool. It's so we actually has a Canadian presence. Unfortunately, yes?
I have a question. So we're talking about carnivorous plants. So do they all eat the same thing, essentially insects?
It's generally invertebrates, predominantly insects. But as we'll see, they've sort of branched out a little bit into catching other things as well. But yeah, generally it's small stuff. This is like I said, Sarracenia. This is about all I'm going to say on it, because these are the pitcher plants I'm going to be talking about. These are the Nepenthes, the Asian pitcher plants, which for my money are the absolute creme de la creme of pitcher plants. But I'm biased. There's about 120 species. When I started working on these just over 20 years ago now, there were 80 species. They haven't evolved that fast. It's just that people have found different new species. Whenever anyone goes up a mountain in the southern Philippines, they come back with generally a new species of Nepenthes. They're hugely speciose on mountaintops. The central diversity of the islands of Borneo and Sumatra, they've got over 30 species each. This is a map of the Indonesian archipelago here. There's Australia, New Guinea, Borneo, Sulawesi, up to mainland Asia. So the stronghold is the center of this area here. There's one species found on Madagascar, or maybe two now, I can't remember. There's Pavillii on the Seychelles. There's a little species in northeast India. Don't even ask me how that got there. But the main area is Southeast Asia. The climbers, they're kind of a funny little plant. Is there a marker pen here? Is that a? I made the mistake of using a non-washable one once, and I'm still hearing about it. Oh, thank you. So what the plants do is they start off on the ground, as most plants do, apart from epiphytes. They produce pitchers which rest on the ground, and as a plant grows, it's a vine, essentially. And they put out a big long tendril which wraps around adjacent vegetation. They produce pitches then, which are aerial, up in the air.
Do they kill the host?
No,
No.
no. As I've got a photograph here. They can. This is a species from Borneo, Nepenthes gracilis. You can see it's on a. This is Pleuarium alternifolium. It's basically covered the host plant, but it doesn't kill it. It would certainly outcompete it to a degree for light.
Are they predominantly terrestrial?
Yeah, there are some which are epiphytic, but not many.
Epiphytic.
Epiphytic means they grow on the branches of trees. So, for instance, a lot of you see a lot of the ferns in the forest here will just be growing on branches of trees. Those are epiphytes. So they're basically rooted in the gumph, the leaf litter, which is on the tops of branches.
So this is a mutualistic relationship?
No.
Mutualistic is, can you differentiate for me the ones that don't kill and the ones that do kill the host?
Yeah, for instance, something like a strangler fig would start off as a seed, a hornbill or something like that would poop in a treetop. The seeds would germinate. The strangler fig would then put roots down. It would then enclose the host tree. It outcompetes it for light and kills the tree. And a mutualistic association is one in which you have two partners, different species, both of which benefit from the association. So, for instance, us and our E. coli. The E. coli helps me. I give the E. coli a home. No questions asked. we both do well. That's mutualism. Well, these are just using the plants for support.
Okay.
I mean, they could do the same in a dead tree. They don't get all they get is elevation. Diane.
Jonathan, evolutionarily, why is it Southeast Asia is the location geographically in the world that these plants exist?
I think it's I mean, it's the reason that they're not in the Americas, for instance, or Europe is just that they never got there. The original species from which this group has evolved started out somewhere in the old world. We don't know whether it was East Africa, Madagascar, or whether it was actually Asia. But as the plates move round, and these are obviously millions of years old, as the tectonic plates move round, they got carried. The tectonic plates are just rafts, so you get some stuff going to India. I think that's why you've still got that stuff in eastern India, where it was basically cut off by the Indian plate when it hit. They're basically just rafting on the continents and on the islands, which are following the tectonic drift. I mean, they're all over the world now because people transport them. But naturally, that's the only area. Did I answer your question properly?
Yeah, I just wondered why.
Okay.
You know,
Yeah,
from a
that's-
Darwinian perspective, why?
Yeah,
Why
that's-
are they
that's-
growing in Southeast Asia? And
Well, the thing is, there are pitcher plants all over the world. There's these ones in Southeast Asia. There are the Sarracenias in North America. There are the sun pitchers, the helianthers in South America. There's one species, which is the only species in an entire family called Cephalotus, which grows in southern Australia, which looks very similar to Nepenthes. It's not related at all. It's an example of convergent evolution. There's the same needs drive, or the same opportunities have led to this convergence of traits. They look very, very similar to Nepenthes. They're not related in any way. So yeah, I think this is an adaptive radiation, this particular thing, where one, a very small group has branched out because there have been lots of ecological opportunities presented to it. We're working on that at the moment, but I think that's what it is. I think this is a good candidate for an adaptive radiation.
And it's not highly specialized then?
Some of them are. This one isn't. I'll show you some really specialized ones later. Okay. They grow in nitrogen-poor environments generally. I've got a picture here. This is Nepenthes truncata from the Philippines. I'm a little jet lagged because I had to catch the red eye back from Mindanao this morning. But this is one here. You can see it's an extension of the leaf. This is the leaf coming off the climber. This is the laminar of the leaf, does all the photosynthesizing, and you've got the pitcher at the bottom. This is very, very good at harvesting light. It's a nice flat plate aimed at the sun, does all the right stuff. This is not a very good structure for harvesting light. This is very inefficient. It photosynthesizes, but it's very inefficient. It's expensive to produce. It's full of proteins, it's full of carbohydrates. It's a big hulking structure. This is very expensive. There is no reason for a plant to produce this unless there's a real need to acquire nitrogen from something other than your roots. This is an extravagance the plant can't afford in areas where there are high nutrients. If you try and grow pitcher plants in a greenhouse or something and you fertilize them, you'll get these beautiful green leaves and they won't produce the pitchers. So they grow in nitrogen-poor environments. These are the only environments where they have an edge with other vegetation. It gives them a competitive advantage.
So basically the reproduction is nutrient induced.
Isn't it
If
always?
they get enough nitrogen, they're not going to bother reproducing?
I don't know. I know that I tried an experiment many years ago, but it didn't run for long enough. The species I was growing on was very low, slow growth, and I suspect that's the case. I don't know that. I suspect though. Yeah, there's still plenty of work to be done on these things. It's an open field. They catch small prey items, generally insects, and they get nitrogen from the prey. Ooh. Could you click then, please? Thank you. All right. Just click.
I think you'd be very good at parties.
Oh, actually, I've got it now. What I'm going to do is I'm going to hand round this picture. I'm going to slice it open. I'm going to hand it round. I'll hand one section around this way. I'll hand one section around this way.
Jonathan, this may be a stupid question. It has to be open to catch the insect. This is close.
Yep. Yep. What you'll see in that picture is at the top, actually, you have the open mouth at the top. These are gravity traps. The open mouth at the top produces nectar. There's nectar all over the place, but they produce nectar around the mouth, or the peristome is what's called, by small nectaries. Insects are attracted to that. If they try and, we'll see in a minute how the plant makes them slip. But if it slips in, you see that there's a boundary here between the upper and the lower. This is waxy. This isn't. This is full of digestive and excretory glands. This is usually full to about here with water, depending on the time of year. So we'll just go through a couple of the. Thank you. Just go through a couple of the ways this plant makes a living. The other thing about the plants is they produce two different types of pitcher. This is the lower pitcher. So when the plant starts off, it produces these ones on the ground, or the terrestrial pitcher form. And the same plant will produce a very, very different pitcher higher up when it's climbing. The lower pitchers have these wings, these paired wings on the base. The aerial pitchers don't. So what we're going to look at is how does this pitcher work? If you feel the outside of that pitcher, it feels slightly rough. It feels a bit velvety. And the reason it's velvety is because if you look under a scanning electron microscope on the outside, the outside is covered in these tiny branched trichomes, these tiny hairs. And this is to give insects and other invertebrates a good grip to allow them to climb up the side of the pitcher. It's in the plant's interest to make it easy for animals to get in. The peristome, which is that ribbed rim at the top of the pitcher, this is, it's shaped a bit like, it's called an anisotropic surface. This is a unidirectional surface. There's overlapping cells here, which is a bit like, it looks a bit like roofing tiles. It's very easy for an insect to gain a foothold climbing down into the pitcher, but to climb up, it can't gain a purchase. So the structure is such that it's a unidirectional bearing surface. If you look very, very closely on the inner rim of that picture, you'll see tiny little teeth, and in between those teeth are nectaries. This is a scanning electron micrograph through the peristome, showing the nectary. This produces the sugary secretion which attracts and maintains prey at the mouth of the pitcher. So the prey's happily feeding. If it manages to maintain its grip, it feeds on the nectar. Occasionally, it's going to try and want to get a better access to this nectary, and it'll climb inside the pitcher. It's SOL at this stage because the inside of the pitcher, as you can see from the upper part of that pitcher, is waxy. These are wax crystals on the outside. These are stomatal, or one part of a stomatal guard cell pair. Stomata are the holes through which plants breathe. They have guard cells on either side of the hole that swell or shrink to maintain, to control gaseous exchange and water loss. What they've done is they've taken these breathing holes and they've turned them into something else. They've turned them into a structure which makes it impossible for an insect to grip. So as well as having these ungrippy surfaces, they're also coated with wax in some species. It clogs up the feet of the insect, and the insect will fall into the pitcher. This is a view, an insect's eye view, as it's just lost its foothold on the pitcher. This is the last thing an insect sees before it hits the drink. This is the moment. You can see that these are the teeth of the peristome, and in between these are the nectaries. Once it hits the drink, it hits the water. As you can see on the picture I've just handed around, if you look very closely, there are tiny little spots on the inside at the base of that pitcher. These are secretory and absorbing glands. These are very complex structures. They produce the water in the pitcher. They pump ions in. They suck ions out, and we'll see what they do a little later on. But they basically produce the proteolytic enzymes which break down the prey. They also absorb the nutrients that are produced. I don't know about you, but I hate the fact that whenever you're eating your dinner, they have these acne adverts on TV. Do you really need them when you're eating your dinner? Some zitty kid talking about the skin problems. This always looked to me like one of the shots from this. This is actually a scanning electron micrograph of the inside of a pitcher, again with this huge array of these secretory glands. This is a very, very physiologically active part of the plant. So what does a peristome do? Remember, this is the rim around at the top of the pitcher that produces the nectar. As we've seen, it's got these sort of slidey tiles. It's not a perfect trap, though. You can see this is an insect foot. This is from a person I've worked with before, Ulrika Bauer from Cambridge, and she's done a lot of work on this. The reason I'm using other people's work as well as mine is that my work ties in with other people's, so it's not that I'm running short of material. She did some beautiful work a couple of years ago showing that the peristome either is a very good surface for trapping or a very poor surface for trapping, depending on the amount of moisture available. If you look at these lines here, if this gets wet, capillary action will draw water up these grooves. It's a highly wettable surface. It's extremely slippy when it's wet. And I'm going to show you now some beautiful video that, again, not myself, Ulrika Bauer took. Just give me a second. This is from her work in Borneo. This is Nepenthes rafflesiana. You can see this is... Okay, let's just... When the pitcher is dry, the ants can basically crawl all over the peristome, drink the nectar, and go away home, no problem. Watch what happens when the peristome's wet. The plant turns into a death trap. The term she used was aquaplaning, and I think it's a fairly apt term. Aquaplaning, yeah, they just slip into it.
Is the fact that it's not slippery when it's dry, does that actually aid in capturing more insects because you get the attraction to when it's dry?
Have you read the script? No, I only asked that because that's my next point.
Okay, sorry.
Yeah, no, very brilliant. No, I'm glad you asked. Well, thank you for asking that.
Welcome.
Ants recruit to a food source. The foraging individuals are sterile females. They'll go out and look for food. When they find a food source, they'll lay a chemical trail back to the rest of the crew and bring all the sisters out to go for the food. If these plants were perfect traps, they would only catch as many ants as come across them by chance. Because they're imperfect traps, they catch a hell of a lot more ants than they should. It's very, very, I'd say clever, but it's a leaf, you know.
Would you describe it as almost the perfect killing machine?
No, the Terminator is a perfect killing machine. These are pretty good.
Yeah, I mean,
Okay, so that's the basic functioning of the picture. We'll start looking at some strategies. You've seen all this before, haven't you? I mean, just if I make any mistakes, let me know. We'll start with a simple one. This is the one I showed before, climbing up the bush. This is Nepenthes gracilis, grows in Borneo and lots of other places. It's very simple. It catches mainly ants, fairly unadorned pictures. There's very little difference in what the aerial pictures catch and what the terrestrial pictures catch. It's mainly ants. Yes.
Is there like some kind of mechanism that triggers the pitcher plant to draw water to the rim of the top of the pitcher?
There are a couple of things will do it. Rain will do it. Moisture will do it. So in the early morning, before it's dried off, the plant will just accumulate moisture on the peristome, which will wet it. Nectar will do it as well, and also if it rains, it does it. So
are they constantly full of liquid too?
Well, not full, but there's usually some liquid in them. Yeah. If it's very, very dry, they can reabsorb that liquid to use for other purposes within the plant. So it's also a reservoir for the plant. So that's a pretty boring one. It just kind of catches ants. Sympatric with this, which means it grows in the same place as Nepenthes gracilis, is Rafflesiana. This is a lot more sophisticated. It catches a lot more flying prey. It catches bees, ants as well. It catches flies, moths.
this is a silly question, I guess. Again, do they die slowly or quickly? Or do you know?
Do the pitchers die, or the plants?
No, the prey.
Oh, very quickly. There's a wetting agent in the water, and they also get torn apart by animals that live in the water. I took a bunch of pitchers back to a laboratory once, and it was an air-conditioned laboratory, nice and cold. Left them in a bucket, and during the night, a gecko, one of these little, Marcel lizards, went down for a drink into the pitcher. It drowned, but one of its legs was stuck on the outside of the pitcher because it got these sticky legs.
Oh no.
And when I came back the next morning, I basically had half a gecko. There was a gecko to the waterline and then nothing. So the spine, the skull, the arms, everything had gone overnight. That was a combination of the organisms, maggots and mosquito larvae in the fluid, and also the enzymes. The thing was gone. And that was in air-conditioned laboratory, that was cold.
So they don't get digested in
The
the...
maggots that live in there
Yeah,
are
they-
being digested by the plant.
No. There are lots of organisms which only live in Nepenthes pitchers.
And they reproduce in there?
Yeah, mosquitoes will lay their egg rafts on the surface of the fluid, and a mosquito larvae will go in. There's lots.
Is that fairly common for a lot of pitchers? I know Sarcinias have a little bug that lives in them too.
Yeah. Yeah, it's what's known as a phytothelm, which is an aquatic habitat contained within a plant. And it's, depending on the species, some species will have lots and lots of organisms living in them. Yeah, it's quite a habitat.
Doesn't that undermine the whole reason the plant exists as a carnivorous plant?
Some of the insects will, obviously, if they're feeding on what's gone in, and when they pupate and fly off, like mosquito larvae, for instance, they'll take a lot of those nutrients with them. Yeah, so they are stealing. In some cases, they help the plant, and I'll show you that in a short while. So basically what you find is Nepenthes rafflesiana. These are the lower pitchers, the ones on the ground. The upper pitchers are the big fancy ones up in the air. You can see that there's quite a difference in the prey they catch. So the upper pitchers are catching lots of flying prey, lots of bees, flies, moths, that kind of stuff. Is this just due to position? You know, the upper pitchers are upper. There's probably lots more flying prey above ground than there is on the ground. What I did was, it's a long time ago I did this. God, still had hair. Bloody hell. If you shift them around, you put the lower pitchers up, you put the upper pitchers down, the upper pitchers still catch more prey. There's something going on in the upper pitchers which makes them more attractive. The aerial pitchers, the upper pitchers of this particular species, Nepenthes rafflesiana, produce a sweet scent. It's not a very pleasant sweet scent. It smells like a dead granny. You know, sort of, you know what I mean? Yeah, you know exactly what I mean, right? Yeah. The fluid is what produces this smell in this particular species. If you take the fluid from a fragrant variety of this and put it in a non-fragrant variety, there's a sympatric non-fragrant variety in Borneo. So you take the sweet-smelling fluid, you put it into these non-sweet-smelling pitchers, and they catch a lot more insects. So it's the scent which is bringing a lot of these insects in. The work I did was very, very primitive. I just basically swapped pitchers around. Some people recently from Europe did a lot more sophisticated work, and they actually identified what the compounds were which were producing these smells, and the compounds which are commonly found in flowers. In flower scent. Remember, this is a leaf tip. This isn't a flower. It's a leaf tip which is producing very, very similar properties to flowers for the same reason, to attract insects. The Nepenthes rafflesiana also has a very, very sophisticated visual signaling system. We're trichromatic, which means we see in red, green, and blue. The majority of insects see in ultraviolet, green, and blue. They don't see in red. So the visual acuity is shifted to shorter wavelengths. If you compare the colors of the peristome, ignore the red stripes there. Most insects don't see red. I'm still not sure why these plants do this, but it's not to attract insects with the red stripes. If you take a color scan using a spectroradiometer of the peristome, which is where the nectar is, remember, and the body of the pitcher, you get this contrast pattern. And you can see that the contrast maxima are in the blue and green, and there's also, it doesn't matter whether it's positive or negative, it's in visual contrast is the important thing. Also in ultraviolet. So the peristome is standing out in the wavelengths to which insects are most sensitive. It's producing a signal saying this is where the nectar is. This is a picture. I took it in my linen cupboard using a black light. I don't get out much. What I did was I got a Q-tip and I wiped the nectar. I got distilled water and I wiped the nectar off one half of the peristome where it's dark, and I left the nectar in. What you can see is the nectar fluoresces. The nectar is absorbing ultraviolet and it's pumping out light in the blue part of the spectrum. Fluorescence is merely the situation where a substance gets hit by light of a particular wavelength. In this case, it's ultraviolet, very, very high energy. The energy excites electrons in the electron shells above the ground state. Now, when the electron can't stay up there indefinitely, it drops down back to its ground state, releases energy. Because it's an inefficient system, it releases it at a lower energy state. This is a longer wavelength of light, so ultraviolet light goes in, blue light comes out. So in one move, the plant is making the peristome UV absorptive and also emitting blue light. And it's doing that using the nectar. What this means is, when you think about it, that at the time when the peristome is at its slippiest, where it's coated with this nectar, it's also at its most conspicuous for catching flying prey. Now, if that isn't elegant for a leaf tip, I don't know what is. This is just the effect quantified here. You see it's absorbing an UV, pumping it out in the blue and green. What this means is that, remember that boring one, the Nepenthes gracilus, the one that was all over the tree? So you get the boring one, the exciting one, growing next to each other. The exciting one's catching all kinds of prey that the other one isn't catching by reason of this very, very conspicuous peristome and the fragrance it produces. Okay, now it starts getting weird. This is a species called Nepenthes albomarginata. It produces very, very little nectar. But what it does have, it has this white band that gives it its name, albo marginatus, just means white margin, obviously. Any Romans in the audience tonight? No.
Friends, yes.
Producers. Any countrymen? No. These are often found full of termites. They produce very, very little nectar. The nectaries are almost non-functional in this species. Oh. Okay. Whoa. There's a really cool group of termites called Hospitalitermes. They have little squirty nozzles on their heads. They spray. They basically spray resin on attacking animals. They're really cool. It's cool to prod them and watch them squirt. Again, we don't get out much. Again, like ants, they recruit to a food source. This particular group of termites feeds on lichens. Talking of mutualistic associations, a lichen is a mutualistic association of a blue-green alga and a fungus. That's what these things feed on. What the plant has done is the white band. You can see these little squirty nozzle termites feeding on the white band. It's produced something which appears to the termites to be lichen. And it's using that to attract them. Do you remember if you rubbed the outside of the picture there, it felt velvety, and I showed you those trichomes? If you look at this band, it's essentially these trichomes cranked up to 11. This is a scanning electron micrograph of that white band. You can see all these trichomes. So the interesting part is here, these little threads. These are fungal hyphae. The plant appears to be growing a fungus garden, which is what is attracting, I think is attracting termites. We still haven't published this yet. I need to go and do some more work. But the interesting thing is you can see this effect in plants in the wild in Malaysia. If you take a scanning electron micrograph of those, you'll see this. If you grow a plant in a terrarium in Canada, you'll get the same thing happening. It's probably not, almost certainly not the same fungal species, but the fact is this is a very, very good habitat for fungus. I suspect that the trichomes are maintaining a boundary layer around the plant which is very, very highly moist, which is enabling fungal growth here. This is what's attracting the termites. So it's growing, I think it's growing a fungus garden to attract them. We know that the band is what attracts them, but I think it's the fungus that's the mechanism. Like I said, we still haven't proved it yet. We've got a couple of experiments to do yet. Okay, now it gets a bit stranger. This is Nepenthes ampullaria. The majority of Nepenthes grow in very, very open environments. Nepenthes ampullaria is an anomaly. It grows under closed canopy forest. Now, the main route of nutrient cycling in forest is litter fall. We've seen it at the moment with all the leaves coming down. They've got all the nitrogen in the leaves that they didn't manage to suck back into the trees before they drop the leaves. In tropical forests, the type of tropical forest in which these plants grow is called heath forest. The trees generally have fairly small leaves. And every year, a square meter of forest floor will receive about 5 grams of nitrogen coming down in the form of leaf litter. This is available for anything that can grab it. And this plant grabs it. You can see here there's a leaf has fallen into this pitcher. The Nepenthes ampullaria forms these beautiful carpets of pitchers, and they look like nothing more than a bunch of mouths all aimed up at the canopy. You can see the lids are reflexed back. They don't produce much nectar. And what you find is, this is great in a dark room, but it's not a dark room. But you can see in here, this is absolutely full of little twigs and branches and leaves and all kinds of crap. It looks like somebody's put a bit of chewing gum in here too. So these plants are basically catching leaf litter. But what can you do with leaf litter? Anyone ever tried to chew a dead leaf? Like one of these ones out of the forest? Anyone got really just stoned and just like wonder what they're very indigestible. And this is where the insects that live in the pitcher come in handy, because what they do is they help the plant. They're not helping it because they care about the plant. They're helping it through their own, because of their own needs. But what happens is you get the leaves going into the water. You'll get a film of bacteria which start to break down the leaf. It's called a biofilm. They start to break down the leaf. Mosquitoes lay their eggs, as I said, in the pitchers. the larvae of some species are grazers, and they will graze like cattle on the biofilm of bacteria on these leaves. So the nitrogen goes from the leaves into the biofilm. The mosquito larvae take up the biofilm. They use it. They use the nitrogen, the nutrients to grow, but they also excrete. Everything excretes. They pump out ammonium ions into the fluid, and that's what the plant takes up. And that's we've estimated using stable isotopes that the plant gets up to about 35% of its total nitrogen in the plant is from leaf litter. The rest is from insects falling in as well, but it's definitely moving in a different direction. Now there are some things you have to do if you want to make your picture a friendly environment for these mosquito larvae and things to feed on. You have to make certain changes. This is a section through Nepenthes ampullaria. A, B, C, D, and E are just the various zones here. This is Nepenthes rafflesiana, the one that catches all the moths and all the cool stuff. You can see it's got the big glands at the bottom. Then the glands get smaller, and you get these semilunate cells and the wax cells at the top. So basically the upper two sections of Nepenthes rafflesiana are all to do with catching and retaining prey. Only the bottom is to do with absorption. Look at ampullaria. It's basically almost the same from the top to the bottom. It's all these glands. There are no semilunate cells. There are no waxy layers. There's nothing to trap prey in this. The whole thing is turned over to absorption of nutrients. This means that it's easy. Essentially, the larger a volume of water you have, the more both the more species and the more individuals you can contain in it. It's a larger system, a more stable system. What this thing's done has turned the whole picture into a pond to keep these organisms alive. These are the glands, remember, in the base and also up to the top of this plant. They have two functions. When the picture is just forming, they'll pump the water in, they'll pump the enzymes in. Once a picture's matured, the function changes and they start taking up the ammonium and other things from the fluid. They also do something rather interesting. Well, as if that isn't interesting enough, but this is a fairly low, high pH. It's slightly acid. This is very acid pH here. Basically, don't worry too much about all the gump, but what this says is that when the conditions are very, very acid in the fluid, Nepenthes ampullaria will suck protons out of the fluid to make it less acid. They maintain the acidity at levels at which things like the mosquito larvae can thrive in. Nepenthes bicalcarata is another one which does the same thing, and I'll show you why later. But it's basically it's a balancing act between making the conditions acidic enough that your proteolytic enzymes will be able to break down all the meat you've got in there, and making sure that it's not such a hostile environment that things won't survive in it. What this means is that very often you'll find lots of things poking around Nepenthes ampullaria pictures. This is a photograph I took in Borneo. This is a crab. This orange thing is a terrestrial crab. Many species of crab are actually terrestrial. There are species of crabs in Jamaica which only live in the tops of trees. They don't even come down to the ground. And they're very good at exploiting niches. They do, however, need to keep the gills wet. Remember that these pictures are fluid filled. I suspect what's happening is that these things are foraging on the forest floor, and every now and again they're popping into a pitcher to replenish the gills with oxygenated water. I don't know. Again, this is an ongoing investigation. This is a picture taken in Singapore of a different species. It's the same genus, Geosasama, the crab, dipping into a Nepenthes ampullaria pitcher. So it's not just on Borneo, it's on the mainland. This is a widespread phenomenon. These are very good habitats for different animals to live in. Oh, sorry.
Does the crab, can it get out afterwards?
Yeah.
Or is it stuck in
No, they get out. Yeah, they're pretty big, so they sort of hook the legs over and just sort of whip down. But yeah, they do fine. I've never seen one dead in a picture. The other thing you get is frogs. I wrote a chapter for a book many years ago, and I had a picture on slide. It was this picture. This is a genus of frog called Phyllotus, which is one of the smallmouth frogs. Everybody know the wide-mouthed frog joke? All right. It's a smallmouth frog. They go. Do you want it? You want to hit it? Oh. All right. So this wide-mouthed frog's walking through the forest. He goes up to this lion and he says, "I'm a wide-mouthed frog. What do you do?" And the lion says, "I'm a lion. I eat antelope." "Oh, that's nice." And he walks off and goes up to a snake. He says, "I'm a wide-mouthed frog. I eat flies. You know, what do you do?" And the snake says, "Oh, I'm a snake and I eat birds." Goes to this swamp and he sees a crocodile. He says, "I'm a wide-mouthed frog and I eat flies. What are you?" And the crocodile says, "I'm a crocodile and I eat wide-mouthed frogs." And the wide-mouthed frog says, "Oh, you don't see many of those nowadays." So, back to the this particular group of frogs are very unusual in that you can see that these are froglets in the eggs. Instead of the egg hatching a tadpole and then the tadpole going through metamorphosis outside the egg, it goes through the entire metamorphosis within the egg. This is quite unusual for frogs. This was a bunch of eggs I found in northwest Borneo in a pitcher. So I did this thing in this book chapter, and I'm Googling this genus on the internet a few months ago now. And this picture comes up, and it's my picture, and some swine has scanned my picture from the book in a really crappy way and put it in. So I'm expecting royalties from these people, but I don't think I'll be getting them. But the point is that if the conditions are good enough for frogs to live in, they're pretty benign. Amphibians don't generally tolerate highly acidic conditions, so the species such as Ampullaria and Bicalcarata, where they actively maintain higher pHs, are basically opening themselves up for exploitation by these groups of animals.
What is the pH?
The pH in something like Rafflesiana can go down to 2, 3. In that species, it can go up to about 6. So it's a hell of a range. As I said at the beginning, lots of Nepenthes grow on mountaintops, and one of the problems with mountaintops is there aren't many insects up there. The higher up you go, both the smaller the biodiversity, and also the smaller the actual density of numbers of organisms. So there's less food for these things to eat up mountains. So what the hell are they doing up mountains? Well, it's very, very strong selective pressure to find another way of sequestering nitrogen from your environment. This is a species, Nepenthes lowii, very weird looking pitchers, very, very tough pitchers. They produce huge amounts of nectar on the lid here, and they're often full of crap. And I mean that in a very real way. This is another picture. You can see something's gone number 2 on here. And we found this a long time ago, and it took us a while to get round to figuring out what was happening here. And I've got some video. Again, this is taken by my colleague Ulrika Bauer, who was the only person with the patience to do this. This is Tupaia montana, the montane tree shrew. It's licking all the nectar. It's scent marking with its anal genital glands. But before it did that, it actually had a bit of a poop. And it's scent marking. Oh, it's scent marking because this is a very, very valuable resource. These plants produce huge amounts of nectar. These shrews, tree shrews, among other things, feed on nectar. You can see it's having a really good feed there. Take a quick crap. It's using its anal genital glands to scent mark that, which says this is my plant, stay off. We estimated, again using stable isotopes, that about between 50 and 100% of the nitrogen in these plants is directly derived from mammal feces. So these are very, very, this is a beautiful mutualism, example of mutualism. They're both acting completely selfishly, but each is benefiting the other.
How long does one pitcher last?
That particular species, probably at least 6 months, 7 months, maybe up to a year, depending on how heavily they're used. Oh, sorry. Oh, no. Damn. You hate amateur slideshows? Okay. All right. Here's a picture by one of my colleagues, Chen Li, who spent an awful long time sitting in the rain waiting to take this shot. But you can see that the tree shrew is. Now you can also determine the sex of the tree shrew. Can anybody tell me what the sex of the tree shrew was that used that? It was male. Why was it male? Because the lid's up, exactly. Remember, the peristome is generally a fairly slippery surface. In the aerial pictures of Nepenthes lowii, the peristome has been flattened. You can see this is the typical lower picture. Remember, they produce two picture types. Lowii produces these lower pictures as well, but they have, they catch insects in the usual way. In the aerial pictures, you can see under scanning electron microscopy, these ridges have been flattened down, so the thing can take a good grip while it's reading the newspaper. The world's largest carnivorous plant is Nepenthes raja. Raja means king in Malay. It was named, I suspect it was named after Raja Brook. These are giant, these are like rugby balls, these things. And over the years there have been quite a lot of stories of these things catching rats, which sounds great, but what we think is happening is that rats are actually clumsy tree shrews that slipped while they were taking a crap. Because these do exactly the same things as Nepenthes lowii. They produce huge amounts of nectar. They're full of tree shrew feces. There's another species which does the same thing. This is Nepenthes macrophylla, another of these giant pitchers. This is an area that we're starting to explore a lot more now. I'm going out to Borneo next February to look at the nutritional status of these various tree shrew species, with and without inputs from tree shrew feces, to see if we can see if there's the same thing going on with these nutritionally, or whether it's just the occasional crap the tree shrews are taking. But we think not. This is the last species I'm going to look at here. This is Nepenthes bicalcarata. It's named for these huge big fangs. The fangs are actually giant nectaries, and what happens? This is hollow. A queen ant will dig a little hole into the plant here, make a nest. This is called a domatium. A domatium just means a home. It's a home for ants. The plant basically provides a home. It provides food. These nectaries are very, very productive. You can see the droplets of nectar on here. They produce huge amounts of nectar. They keep the ants very, very happy. What the ants do three things that we know of so far. The ants will keep away weevils, which are small beetles which will eat the leaf tips when they're growing, so they keep them off. If a large prey item falls into the pitcher, like a large cockroach, the ants will dive into the water. These were the first ants that were discovered to swim. They'll pull a cockroach out, chew it to pieces, and throw it out of the pitcher. It stops the pitcher going anoxic. It basically stops the pitcher being killed by the oxygen demand of a large piece of meat going in. And the third thing is they also poop. I'm going to be working next spring with some French researchers out in Brunei to investigate whether there's actually a nutritional advantage to these plants from having these ants.
How big is that one?
Like that.
And do the ants live exclusively on
Yes,
these? Like, is this
the ants
their-
are found nowhere else. They're completely dependent upon this species. Ooh. All right. Little light reading for you if you want.
Thanks.
I just want to sort of finish up before we take any questions. Just want to say thank you very much for coming and showing an interest in this rather esoteric topic. I'd also like to thank the research office for supporting me for the last few years. Like I said, this is kind of the stuff I do off the side of my desk. The main stuff I do is the hummingbirds, but there are so many very, very interesting questions still to find out today. So I'm very, very thankful for all the support I get from the research office. Thank you for this. And any questions? Anne?
Jonathan, given your knowledge of of the adaptation to these plants, and, you know, it is kind of a brilliant killing machine. Are there any lessons for how we humans organize ourselves and our society? Any sort of application from your research to, it's kind of a convoluted question, but figure out where I'm going?
As I said, they're not completely efficient. A lot of animals that visit these plants get away with feeding and don't get killed. So I suppose the thing is, if you're going to do anything, in many cases, I think it's more of an exchange than a true predation. In the ants, for instance, it's sterile females which are trickling into the picture. The queen isn't harmed. I think there's a net advantage to these plants, to the ants, for associating with these plants. So yeah, they're getting preyed on, but because they're modular organisms, they're not losing anything they can't replace. And in many areas, the pitcher plants are the main source of nectar year-round. They produce nectar year-round. A lot of the other flowering plants don't do that. You could look at it in the same way as, for instance, a mutualistic association of the bull's horn acacia with the Azteca ant in Central America. They protect the bull's horn acacia produces these big thorns. The ants live in them. The ants protect the plants against cattle and grasshoppers and things like that. A lot of the ants will die during the defense of the plant, so that the modular organism that is the ant nest is losing some of its modules, but it's getting a home, it's getting nutrition. I suspect it looks like a similar thing. So I think if there's a take-home message is you don't need to kill everything. You can get away with a little bit of kindness as well.
Generosity.
Thanks, Jonathan. How do you design the experiments for these nutrient flux tests? Because with rain, there must be so many variables. Do you move it into the lab, or is it actually field-based?
You do field-based. With the first thing I did was I figured out whether I could do it without damaging the plant. So I did an experiment a long time ago with Nepenthes rafflesiana, where I found a cohort of plants which were fairly young in a fairly discrete area. Half of them I took the prey contents out, put distilled water in and cotton wool to stop them catching any more. And I left them for several months. I went back, I harvested the plants, looked at what the nitrogen and phosphorus in the plants were. At the same time, I took scans of the leaves with a spectroradiometer, and I worked out, using sensitivity analysis, I worked out which wavelengths were the most sensitive to this, to being starved, essentially. That was the only stress the plants had, so I knew it was starvation stress. Once I have those sort of hyperspectral indices worked out, I can then go basically anywhere with these plants, and I can look at relative nutritional status between plants. With, for instance, what we're planning to do with the tree shrews is we're planning to just net some of the bags. So all the other variables are the same. We net the bags, it stops the tree shrews getting in. And then we have someone set that up ahead of time. We go several months later. I take the scans because we're working in national parks, and you can't take. We're working in national parks this time. You can't take samples. I wouldn't want to do that from a national park anyway. So we're having to use secondary methods, but we've got the calibration based on empirical experiments we did previously. What we did with the Lowii was we had permission to take samples from there. We looked at nitrogen 15. We used nitrogen 15 increases up trophic levels to a tune of about 3 parts per thousand. So you can get a good idea of the source of the nitrogen. And using a mixing model, fairly simple mixing model, we estimated between about 57 and 100% of the nitrogen in those plants was coming from the tree shrews. But there are indirect, like I said, if you use reflectants, foliar reflectants, that's a fairly good technique. That's what's commonly used in remote sensing. They do the same thing. They develop hyperspectral indices. So that's what we did.
I have an anecdotal comment on the efficiency of Venus flytraps. We got a present in April, May from someone, a little Venus flytrap, yay big, put it into our kitchen window. It's sitting in a pot with water, so there's always some humidity there, but not directly in the water.
Yep.
We had no fruit flies this summer. We were absolutely spared fruit flies. They were all in this plant. It was littered with little fruit flies,
Nice.
a couple of wasps, a couple of flies, but wiped out fruit flies. So it's a recommendation, actually.
And it's organic.
It's fantastic.
Yeah.
I also did the same thing. I put it beside my organic
safe, and
Yeah.
it's doing great.
That's a great idea.
Yeah.
Nice. Yeah, thank you. Any other questions?
Is there any sort of mycorrhizal activity associated with these plants, or do they grow in association with the host plant, that there's any sort of root contact?
There is some mycorrhizal. My wife did some work years ago in West Malaysia, where she found mycorrhizal associations in the roots of Nepenthes grassless, but we didn't take it any further because we were just leaving. So there's an awful lot to find out about these plants that we certainly haven't. I think we've just scratched the surface, really. Any other questions? Well, thank you very much for coming, and I hope you're thoroughly confused as I am now.
Thank you.